Hi everyone.
Hi
Today
everyone,
I'm going
I'm
to
Saranya
explain
Duraksamy
my project,
and my final project title
predicting
is Big
genetic
Data Case
variance
Study in
via
Life
machine
Sciences.
learning
So
algorithm.
for my case study, I built a machine learning algorithm that will predict genetic mutations. So I'm going to explain one by one slide and I'm going to provide some more information and I'll also show a mini demo. And let's see. So, genetic studies and testing have transformed treatments of many genetic disorders, including cancer. And once a gene is sequenced, a tumor gene is sequenced, it can have thousands of genetic mutation. But currently, the challenge we are facing is distinguishing the tumor causing and the normal mutations. So, interpreting mutations manually is a time consuming task, which calls for the development of a machine learning algorithm which will be able to classify genetic mutations based on clinical information or clinical evidence. So, dataset. The dataset, a genetic mutation can be classified under 9 classes, and for this particular project, the dataset includes both training and test data files for the variations and clinical evidence. So, the training and test variants provides the information about the genetic mutation, whereas the test text or else the clinical evidence provides the information or the clinical evidence that are used to classify the genetic mutations. So, the primary key or the common field that are used to link the text or the clinical evidence and the variation file would be the ID field, which serves as the primary column. And I have provided a link to the dataset if you want to download the data and run the code. So the technology, so the machine learning models were used using both R and Python. So for R, I have used the XGBoost package, and which was used to develop the model. And for Python, I have used the Keras package or the Keras module using which I developed the machine learning algorithm. So some of the techniques that I have included or implemented in this project would be so importing data files and natural language toolkit to remove stop words and like sparse items from the data set, and developing the machine learning algorithm using different models. And so XGBoost would be R. So and I also included a word cloud to identify the most frequently or commonly used words in the clinical evidence. And then there is also a graph using the matplotlib library to visualize loss and accuracy. So going detail into the data format. So the data files are both comma separated. So they are like, so there are 4 data files and 2 are comma separated files and 2 are like, double pipe limited, delimited files. So to explain more about like the data and variables, so the ID field would be the ID of each and every row, which is the unique or the primary column, and the gene is the gene where the genetic mutation is located, and the variation column explains the amino acid change for a particular mutation. So each and every human being has a particular amino acid sequence, but if one word or one alphabet changes in the sequence, that's what causes a mutation. And class would be anywhere between the numerical value for the class should be 1 to 9, and under which the genetic mutation has been classified. And the text is nothing but the clinical evidence that is used to classify the genetic mutation. Okay. So for this particular demo, if you want to run this code, mainly you would have to have R and RStudio installed on your laptop. And so there are like some packages mentioned. So you would need ggplot, ggthemes, and the main package, other packages you would need would be XGBoost, StringR, WordCloud, ggplot, tidyr, and readr. So these are the main packages that you would be needing if you need to run this code. So let me see if I can quickly open my R code. Okay, so as you see in this code, so first I set up my working directory space, and then I load all the packages that I need for running my script. So the first would be loading the data sets. and I upload the train text, which is my training data set, and then I upload my test data set. So once I have my data files upload, I have this, I'll run this quickly. And then I proceed with the language processing methods where there is some data cleanup included. So in this particular text of code, so what I aim to do is I have to remove the trailing space, white space, and then I have to transform all the lowercase. I have to transform all the words into lower cases and then remove the punctuations and remove stop words, which are these stop words that are identified in the clinical evidence, and then I remove all the numbers. So, and then I create a document term matrix. And then once after I create the document term matrix, then I convert that. I then calculated the word frequency to find out the most frequent words. So the most frequent words would be, as you see in this graph, the most frequent words in the clinical evidence were cell and then mutant, which is not that surprising. Okay. So once after I created the bar chart, I moved to go and create the word cloud to see if the bar chart results match the word cloud results. And again, you can see the most frequent words in the clinical evidence were like some of the most clinical. Cell, mutant, tumor, gene, domain, patient, cancer, and active were some of the most frequently used words. And so going to the most important part, model. Yep. Okay. So my data set has 3321 observations for which I divide the data set into test data and train data. So for my train data, I take the observations from 1 to 3000, and for my test data, I take the observation from 3001 to 3321, which would be like 321 observations for my test and 3000 for my train. So I run the XGB model. So initially, the data set is framed into a dense matrix or a D matrix to identify the complex relationship between the features and the labels. So the model was trained using the trained data set, and the class of the test data set was predicted by applying the XGBoost model. And the model was run thrice to predict the value of the class variable, and it found the mode of all the three results obtained, which decided the final value of the class. So the XGBoost uses a tree learning algorithm where, if you see here, the verbose value is set to 1 in the first two nodes to print the evaluation metric, which enables us to check the progress of the training process. And here, the verbose is actually set to 2, if you see. And so this is to print the tree information, details of which I have included in my final report. So I ran the model for both the test data set and the training data set. And once after I ran the model, so this is for the training data set, and then I repeat the same steps for my test data set. And finally, once after I finish running the model, I have so this is what the XGBoost model predicted for me. So for this, you can identify, and the training accuracy of the model was 31.78%, and the final results with which the model predicted were. So you see the ID 0. So for ID 0, the mutant, it belongs to the class 7, and ID 1 belongs to class 4. So it predicts which of these mutants or which of these variants belong to which class of mutants. So again, this is predicted using the XGBoost model, which we are going to compare it with the results of the Python Keras model, which I'll explain in a minute. So I have included my Python script as a Python Jupyter notebook, for which you need to have Jupyter Notebook installed in your system. So just go to command line prompt, type Jupyter Notebook, and your kernel. So the localhost should you should be able to load it into site. And this is my Python Jupyter Notebook for the Keras model. So again, I read the data files into the model. So sorry, and So here you can see the variant info, the training info, and so the test variant info, and then the length of the variant gene. And so I'm directly moving to the model, which is okay. So I import the Keras package, and so you have to previously have it installed. And so once before loading the data, we clean the data using the doctor vector, which converts the words in a sentence into a vector. So after feeding data to the doctor vector, we must train the model. So the model is usually better trained if in each training epoch the sentences or the sequence of sentences fed to the model is randomized. So the model is trained here for 5 epochs and the model is saved to avoid retraining when it's loaded again. I also create a NumPy array as the classifier. The sklearn.model takes only NumPy arrays. So moving to the neural network model Keras. So a little bit of background info about that. So Keras is a high-level neural networks API that's written in Python, and the core data structure of Keras is a model, a way to organize layers. So you can stack layer, and you can create additional layers. I chose the sequential model, which is a simple model with a linear stack of layers. And so here I define the model, and then once after you define the model, you compile the model, and then you fit the model. Yes. And so here we achieve the training accuracy of like 56.63% using the Keras model. And so this is the graph that we obtained from Matplotlib that shows the model accuracy and model loss. So moving on to the final results. So the training accuracy using the XGBoost model was 31.78%, whereas the training accuracy that I achieved using the Keras model was 56.63%, and the validation accuracy using Keras model was 35.94%. So one of the reasons why the training accuracy would be less as the data set is like relatively small. So if we have more clinical evidence, we could create a bigger, yeah. So now we are comparing the results that we got from the predictions of the XGBoost model and the Python model. So if you see here for ID 0, R has predicted class 7, which you can see the same in Python, 0.65 that has the highest value, and ID 1 class 4, and here ID 1 has like 0.7. So the prediction results of almost both the models are same, except that the accuracy is different between both the models.